Right now, as you listen to this, your brain is actively suppressing the urge to, scratch your nose or
Well,
check your phone.
stretch your legs. Yeah.
Exactly. Or just up and walk away. I mean, you have 1,000 competing, entirely incompatible impulses firing at this exact millisecond.
You really are a walking paradox of choice.
Yeah. So out of all those possibilities, how does your brain pick just one? How does it silence the 999 other impulses and commit to a single behavioral output?
That is the big question.
It is. Welcome back to our ongoing deep dive into the neuroscience of movement. For those of you following along with our overarching series, we've been methodically working our way through the complex systems that let us interact with the physical world.
Right. And we're taking our time with it.
We really are. We are currently in part 5 of this journey, and today is a major milestone. We are unpacking chapter 38 of our source material, which focuses entirely on the basal ganglia.
Or, if you might see it in some of the foundational literature, the nucleus database.
Yes, exactly. And our mission today is to treat this like a premium masterclass. We want an unhurried, really deliberate pace so we can let these complex cellular architectures just breathe.
Which they need to, because the basal ganglia represent one of the most intricate, elegant, and functionally dense regions of the central nervous system.
I mean, they're nestled so deep within the brain, right?
Yeah, right down on the core. And historically, the conversation around them has been, well, heavily skewed.
Skewed how? Like misunderstood?
Totally. The traditional clinical view painted these structures almost exclusively as a purely motor center.
Because of the diseases associated with them.
Exactly. When things go wrong here, like, say, in conditions like Parkinson's disease or Huntington's disease, the most obvious visually striking symptoms are profound movement disorders.
Right. The tremors, the rigidity, it's what you see first.
Yeah. So those clinical observations drove a very narrow physiological assumption for decades. People just assumed these nuclei were deep brain relay stations sending projections up to the motor cortex to help us, walk or reach or grasp.
I want to make sure we don't just gloss over that historical bias, though, because it makes total sense why early neurologists would think it's just a motor center.
Oh, absolutely. It's a completely logical leap.
Right. If a patient comes in with a severe tremor or an inability to initiate a step, and then an autopsy later reveals profound damage in the basal ganglia, you'd think, well, this part of the brain controls movement.
You would. But we need to completely reframe that paradigm right now.
Because it's fundamentally incomplete.
It's a fundamental misconception, yeah. Through modern anatomical tracing and sophisticated physiological recordings, plus advanced computational modeling, we see that the basal ganglia process much more than just motor commands.
So what else are they doing?
They process cognitive, affective, and motivational signals. And they do it using the exact same architectural logic that they apply to motor signals.
Wow. So it's a universal processor for choices.
Exactly. They are not just the brain's motor output relay. They are the brain's action selection engine.
Action selection engine. I like that.
It's the evolutionary solution to the problem of having a highly complex organism with multiple desires, but only one physical body to act them out with.
Right, because you cannot simultaneously walk forward and walk backward.
No, you can't. And you can't simultaneously express intense joy and profound grief in the same behavioral channel.
So it's essentially a bottleneck problem.
Yes, a massive bottleneck.
The cerebral cortex is this huge idea generator, right?
Yeah.
Constantly bubbling up with plans and desires. But the physical body is the bottleneck.
Right. And the basal ganglia are the central clearinghouse. All these competing requests from the cerebral cortex are evaluated there.
They're weighed against each other.
Yeah, and ultimately, that's where a winner is selected.
Okay, so to understand how that selection actually happens, we need to map out the hardware. We have to identify the cast of characters, so to speak.
And the architecture here is remarkably specific.
Let's break it down. I know the source material divides them into 3 categories.
Right. We can broadly divide the basal ganglia based on their function within the circuit. So first you have the afferent centers.
Which are the receivers.
Exactly. Then you have the efferent centers, which are the broadcasters. And finally, an intrinsic center that acts as an internal regulator, and it never talks to the outside world.
Okay, let's anchor this spatially for a second. We're talking about structures deep inside the brain, beneath the convoluted surface of the cortex.
Deep subcortical structures, yeah.
I really want to build a 3D map in my head. Let's start with the receivers, the afferent centers. Where are the signals coming from, and where do they land?
So the largest and most prominent entry point is the striatum.
The striatum.
Yeah. Anatomically, the striatum is a collective term for two major structures. That's the caudate nucleus and the putamen.
Okay, caudate and putamen. And it's huge, right?
In terms of volume and connectivity, it is absolutely massive. It derives its name, the striatum, from its striped appearance.
Oh, like striated muscle.
Similar concept, yeah. The stripes are created by the white matter tracts of the internal capsule passing right through it.
Got it. So what is this massive structure receiving?
The striatum receives direct, topographically organized afferent input from virtually the entire cerebral cortex.
The entire cortex?
Almost all of it, yeah. It is also receiving significant inputs from limbic structures, like the amygdala and the hippocampus.
So emotional and memory inputs, too.
Exactly, as well as indirect input from the brainstem via the thalamus.
So it's like a massive corporate inbox. Every single department in the cortical building, you know, the motor planners, the emotional centers, the sensory processors, they are all sending their emails down into the striatum.
And those emails are glutamatergic excitatory signals, yes.
But the striatum isn't the only inbox, is it?
No, there are two other vital receiving centers. We have the subthalamic nucleus, or STN,
STN, okay.
which receives its own highly organized inputs from the cortex, particularly the frontal lobe, and from the thalamus. And finally, we have the dopaminergic center. This comprises the ventral tegmental area and the substantia nigra pars compacta.
VTA and SNC, that's a mouthful.
It is, but they are crucial. This region receives widespread inputs from the brainstem, the amygdala, and the frontal cortex, and it's going to play a massive role in how the basal ganglia learn, which we will dissect later.
Okay, so just to recap, we have our receivers, the striatum, the subthalamic nucleus, and the dopamine center.
Correct.
They are listening to the rest of the brain, taking in all these requests and contextual clues. Now, on the flip side of this engine, we have the efferent centers, the broadcaster.
The exits.
Right. These are the structures that actually send the final decision away from the basal ganglia.
Yeah, and the two principal efferent nuclei are the internal segment of the globus pallidus.
Abbreviated as GPI.
Yes, GPI. And the substantia nigra pars reticulata, or SNR.
GPI and SNR.
These structures take the highly processed information from within the basal ganglia and project it outwards. Their primary target is the thalamus.
Which then relays the signal back up to the cortex.
Exactly. They also send projections down to vital motor centers in the brainstem, like the superior colliculus for eye movements and the pedunculopontine nucleus for postural control.
Wow, okay, so they're sending signals up and down.
Yeah.
But we missed one category. Sitting right in the middle of all of this, essentially talking only to itself, is the intrinsic center.
Right, the external segment of the globus pallidus, the GP.
GP, so it's the external version of the broadcaster we just talked about.
Geographically, yes. But functionally, the GP is a fascinating structure. It does not receive signals directly from the outside world of the cortex.
Okay.
And it does not broadcast out to the thalamus. It is an entirely intrinsic node, modifying the flow of information between the receivers and the broadcasters.
So structurally, you can picture this entire system as a massive selective funnel.
That's a great way to visualize it.
Information pours in from the expansive cortex into the massive striatum and STN, gets heavily processed through this intricate internal network involving the GPE, and then funnels out through the much narrower exit of the GPI and STN.
Exactly. You start wide and end narrow.
I'm visualizing the macrostructure, the funnel, but the source material spends a huge amount of time zooming in on the cellular level, particularly inside that massive inbox, the striatum.
And it has to, because it seems like this is where the actual magic of the basal ganglia begins.
We have to talk about the medium spiny neurons.
We do. Over 90% of the neurons in the striatum are medium spiny neurons, or MSNs.
90%, that's almost the whole thing.
Yeah, it's overwhelmingly MSNs. They get their name from the incredibly dense array of dendritic spines covering their branches.
Which is where they receive all his emails, right?
Exactly. That's where they receive those millions of glutamatergic synapses from the cortex. And here's the foundational characteristic we absolutely cannot gloss over.
Okay, what is it?
These medium spiny neurons are GABAergic. They are inhibitory.
Hold on, I need to make sure I have this straight.
Go for it.
The cortex is sending excitatory signals. It's shouting, do this, go, using glutamate.
Right.
The medium spiny neurons receive that excitation. They fire an action potential, but their output is inhibitory. They release GABA.
Yes.
So they are essentially taking an excitatory cortical command and translating it into a quiet down signal for the next neuron in the chain.
That structural flip is the basis for everything the basal ganglia do. If you don't grasp that, the rest won't make sense.
Excitatory in, inhibitory out.
Got it. And it gets more intricate because these medium spiny neurons are not a single uniform population.
They're not.
No, they're meticulously divided into two distinct groups. Physically, they look identical under a standard microscope. They sit side by side, totally intermingled in the striatum.
But molecularly?
Molecularly, they are completely different. They are defined by the specific type of dopamine receptors they express and the neuropeptides they co-release.
Okay, so we have two specific flavors of MSNs. Let's break down the chemistry here, because the reading showed that this dictates the entire wiring diagram we are going to look at next.
Right. The first population expresses D1 dopamine receptors.
D1?
Yeah. And alongside releasing the inhibitory neurotransmitter GABA, they also co-express the neuropeptides substance P and dynorphin.
Substance P and dynorphin. Got it. And the second group?
The second population expresses D2 dopamine receptors, and they co-express the neuropeptide enkephalin.
Okay, D1 and D2. But if I'm a neuron in the cortex, right, and I'm sending a signal down to the striatum, do I care if my signal hits a D1 neuron or a D2 neuron?
That's the thing. The cortex doesn't differentiate in its broadcast.
Yeah.
It basically bathes both populations in glutamate equally.
It just shouts at everyone.
Right, but how those neurons actually respond to that glutamate is heavily dictated by the dopamine tone in the striatum.
Ah.
And this brings up a crucial neurobiological reality about dopamine in this system. It does not operate like a traditional point-to-point synapse.
Right. This was one of the most counterintuitive things in material for me. Usually when we picture neurons talking, we picture a wire.
A direct connection, yeah.
Exactly. One axon terminal docks precisely onto one dendritic spine, releasing neurotransmitters across a microscopic synaptic cleft. It's a direct private phone line.
Right, very one-to-one.
So if dopamine is coming up from the substantia nigra pars compacta to modulate this massive inbox, I naturally assumed it was making billions of private phone calls to all those MSNs.
But it doesn't. The dopaminergic projections from the midbrain do something entirely different. They utilize what is known as volume transmission.
Volume transmission?
Yeah. When those dopaminergic axons reach the striatum, they branch extensively. And along those branches are thousands of varicosities, which are basically swollen release sites.
Okay.
And these varicosities often do not make tight, direct synaptic contact with a specific spine.
Wait, so they are just dumping dopamine into the void?
Basically, yes, they release dopamine into the extracellular fluid. The dopamine molecules just diffuse outward, physically washing over a relatively large volume of tissue. So this single release event can interact with the D1 and D2 receptors on thousands of different medium spiny neurons simultaneously.
It's a sprinkler system.
A sprinkler system. That's a perfect analogy.
Instead of watering a single blade of grass with a microscopic eyedropper, the dopamine neurons are a sprinkler system going off, changing the moisture level of the entire lawn.
Exactly. Because it's a volume transmission, the temporal and spatial dynamics are totally different from glutamate.
How so?
Well, glutamate provides a sharp, discrete, millisecond-fast packet of specific information, like move the index finger.
Right. Very fast, very specific.
Dopamine, on the other hand, provides a slower, longer-lasting, spatially broad modulatory tone. It sets the baseline state of the striatum.
It creates the mood.
Exactly, the mood or the context. And that context dictates how sensitively those D1 and D2 neurons will react to the sharp glutamatergic commands from the cortex.
Okay, I think I'm really picturing this now. We have our structural map. We have our D1 and D2 sprinkler-regulated neurons. Now we have to look at how they actually connect.
The wiring diagram.
Right. How do we get from this massive modulated inbox to an actual decision?
For decades, the foundational model for understanding these circuits was proposed by Roger Elbin, Milan DeLong, and their colleagues back in 1989.
The 1989 model.
Yeah, and it's a highly elegant, deeply influential model that has pretty much anchored neurological education ever since.
What did they propose?
They proposed that the flow of information through the basal ganglia is strictly segregated into two parallel competing pathways, the direct pathway and the indirect pathway.
Let's trace these pathways meticulously, because I realized reading this, if you don't understand the routing here, none of the clinical diseases later on will make any sense.
No, they won't. It's the core logic of the system.
So let's start with the direct pathway.
Okay, the direct pathway originates with that first population of medium spiny neurons, the ones expressing the D1 dopamine receptors.
The D1 MSNs.
Right. These D1 neurons receive their excitatory glutamatergic input from the cortex. And when they fire, They send their inhibitory GABAergic axons directly to the efferent nuclei, the GPI, and the SNR.
Hence the name, direct.
Exactly. It's a direct shot.
Cortex excites the D1-MSN. The D1-MSN directly inhibits the broadcaster.
Got it.
What is the functional result of this specific chain of events? Like, what does it actually do?
The functional consequence of activating the direct pathway is the promotion of behavior. It facilitates the execution of whatever cortical request initiated the signal.
Okay, so it promotes action.
Yes. We will dive deep into the biophysics of exactly how it promotes that behavior when we get to the concept of disinhibition. But functionally, for now, you can think of the direct pathway as the brain's ultimate go signal for a specific action.
The go signal. Got it. Now, what about the other side of the coin? The indirect pathway. I'm assuming it takes a detour.
It takes a significant detour, yeah. And it involves completely different structures.
Hey, walk me through it.
The indirect pathway starts with the second population of medium spiny neurons, the ones expressing the D2 dopamine receptor.
The D2-MSNs.
Right. When these D2 neurons are excited by the cortex, they do not project to the exits.
Where do they go?
They send their inhibitory GABAergic axons to our internal intrinsic middleman, the external globus pallidus, the GP.
Okay, so the D2-MSN inhibits the GPE. Where does the signal go from there?
Well, the GP, which is also filled with inhibitory GABAergic neurons, sends its axons to the subthalamic nucleus, the STN.
The receiver we mentioned earlier.
Exactly. Now, the STN is unique here. It is the only purely excitatory structure within the core basal ganglia network.
The only one.
Yeah, it uses glutamate instead of GABA. And finally, the STN sends its excitatory axons to the efferent nuclei, the GPI, and the SNR.
Okay, wait, let me map that out loud to make sure I have it.
Go ahead.
Cortex excites the D2-MSN.
Right.
The D2-MSN inhibits the GPE.
Yep.
The GPP normally inhibits the STN, but because the GPP just got inhibited, the STN is freed up.
Exactly. It's disinhibited.
Yeah. And once it's freed up, it fires its excitatory glutamate down onto the final broadcasters, the GPI and SNR.
You nailed it.
It's a chain of double negatives.
It is a complex polysynaptic chain, but the ultimate result is really clear. Because of that extra layer of routing and the inclusion of the excitatory STN, the functional consequence is the exact opposite of the direct pathway.
So if direct is go,
Activating the indirect pathway acts to fiercely suppress behavior. It's the no-go signal.
okay, Albin's 1989 model gives us this beautiful intuitive balance.
It really is elegant.
The direct pathway is like a sniper rifle promoting a very specific selected action, while the indirect pathway is a shotgun suppressing all competing unwanted actions. It feels very clean.
Almost too clean for biology.
Right. Biology is rarely that tidy.
And it isn't here either. It's a foundational heuristic. But as modern anatomical tracing techniques have advanced, we've realized it's not the whole story.
So what's the modern view?
The basal ganglia are not just two clean, isolated highways operating in a vacuum. The modern view embraces a much higher degree of interconnectivity. For example, we now recognize the critical role of the hyperdirect pathway.
Hyperdirect. That sounds like a bypass mechanism.
That is exactly what it is. The hyperdirect pathway consists of excitatory projections straight from the cerebral cortex that completely bypass the striatum and project directly onto the subthalamic nucleus.
It skips the massive inbox entirely.
It skips the processing delay of the striatum completely.
Wow.
The cortex immediately excites the STN, and the STN immediately excites the efferent nuclei, the GPI, and SNR.
Because it involves fewer synapses, this pathway must be incredibly fast.
Insanely fast.
Yeah.
Physiologically, it is thought to provide a rapid global suppression of movement, a sort of neurological emergency brake.
An emergency brake.
Yeah, that engages before the slower, more highly calculated direct and indirect pathways have time to process their detailed selections.
So if I'm reaching for a cup of coffee, right, and I suddenly see a wasp land on the rim, I don't have time to run a complex cost-benefit analysis through my striatum.
No, you'd get stung.
Right. I need an immediate global stop command. So that's the hyperdirect pathway firing, slamming the brakes on all motor output until the rest of the basal ganglia can figure out a new plan.
Precisely. It freezes you. And on top of that, we also see immense crosstalk that defies the strict parallel pathways of the old model.
Like what?
Well, we now know that the D1 medium spiny neurons of the direct pathway frequently send collateral axon branches to the GPP on their way to the GPI.
So they're talking to the middleman, too.
Exactly. And the GPP doesn't just project to the STN, it projects backward to the striatum and directly forward to the GPI.
It's all connected.
Yeah, and the subthalamic nucleus is no longer viewed just as a simple relay in the indirect pathway. It acts much more like a central associative hub, receiving broad input and broadcasting widely.
So the wiring is less like parallel highways and more like a highly interconnected recurrent neural network.
Very much so.
But even with that localized complexity, there is a massive architectural question we haven't addressed yet.
The macro scale loop.
Yes. The cortex sends an unimaginable amount of data down into these deep structures. The basal ganglia do their processing, weigh the direct and indirect pathways, and then they send the result back up to the thalamus, which relays it right back to the very same cortex that started the whole process.
It loops back.
Why? Why build a brain where the cortex essentially talks to itself through this elaborate deep brain detour?
That reentrant loop is the signature of the basal ganglia.
But what's the point of it?
Think about the cerebral cortex.
Yeah.
It is a phenomenal generator of complex representations, plans, and associations. But structurally, it lacks the dense lateral inhibitory networks required to cleanly resolve fierce competition between entirely different types of ideas.
It can't make up its own mind.
Basically, yeah. It needs an external arbiter. So the cortex presents its menu of options. And these options travel down into the basal ganglia through distinct parallel channels based on their origin.
Meaning the different departments in the cortical building have their own dedicated processing lines in the basal ganglia.
Yes. The basal ganglia are not a single monolithic processor. They are a collection of specialized processors running the exact same algorithmic software on different types of data.
Okay, how many processors are we talking about?
We generally map these out as parallel segregated loops.
Let's walk through those loops, because this is where the action selection idea really expands beyond just moving muscles.
Right. The most well-known is the sensorimotor loop.
The physical movement one.
Exactly. This originates in the primary motor and somatosensory cortices, projects down into the putamen, passes through the corresponding territories of the GPI, and returns via the motor thalamus back to the supplementary motor area.
Okay, so this loop selects physical movements.
Yes, but right next to it, maintaining strict anatomical segregation, is the associative loop.
Associative, where does that start?
This originates in the prefrontal cortex and parietal cortex, projects to the caudate nucleus, and loops back to the prefrontal cortex.
The prefrontal cortex is the seat of working memory, planning, and executive function.
Exactly.
So this loop isn't selecting a muscle movement, it's selecting a thought.
It is mediating cognitive selection. It decides which plan to hold in working memory and which distracting thoughts to suppress.
That is fascinating.
And finally, we have the limbic loop.
The emotion loop.
Right. This originates in the amygdala, hippocampus, and orbitofrontal cortex, projects to the ventral striatum, specifically the nucleus accumbens, and loops back to the anterior cingulate cortex.
And what is it selecting?
This loop mediates emotional and motivational selections. It decides what you care about in any given moment.
It evaluates desires.
Yes.
So I have a motor loop figuring out how to move my hand, an associative loop figuring out the spatial puzzle of a chessboard, and a limbic loop feeling the intense desire to win the game.
All running at the same time.
All three are running simultaneously through parallel funnels in the basal ganglia, using the exact same D1 and D2 receptor logic, and sending their resolved, filtered decisions back to their respective cortical origins.
That segregation is vital. It maintains the specificity of the signal while applying a universal selection mechanism.
But that still leaves us with a huge mechanical question. What is that universal selection mechanism?
How does it actually choose?
Right. We see the wiring, we see the loops, but how does this intricate web of GABA and glutamate actually enforce a choice?
Which brings us to the core neurophysiology of the engine. We need to explore the evolutionary brakes and this deeply counterintuitive concept of disinhibition.
Disinhibition. Okay, let's get into it.
To truly appreciate this mechanism, we have to look at it through the lens of evolution and Charles Sherrington's concept of the final common pathway.
Sherrington.
Yeah. Sherrington articulated a brutal reality of biology. Organisms are fundamentally constrained by their physical bodies.
Meaning, you can have 1,000 thoughts, but you only have 2 legs.
Exactly. The sensory systems have massive bandwidth. You can simultaneously process the visual input of a predator, the auditory input of a rustling leaf, the interoceptive feeling of hunger, and the temperature of the air.
The brain takes it all in.
The brain can model all of these simultaneously, but the motor system is a severe bottleneck. You cannot act on all those stimuli at once in mutually exclusive ways.
Right.
You cannot initiate a stalking motor sequence to hunt prey while simultaneously initiating a frantic fleeing sequence to escape a predator.
Your body would just tear itself apart.
You would just fail. So the nervous system must have an incredibly robust, infallible mechanism to choose one behavioral state and actively, violently suppress all the others.
And this isn't just a human problem. This isn't about higher order thinking. The source material points out something that completely blew my mind.
The lampreys.
Yes. This exact architectural solution has been conserved for over 500 million years.
It is one of the most stunning examples of evolutionary conservation in all of neurobiology. If you examine the nervous system of a lamprey, which is a jawless fish that is one of the most ancient vertebrate lineages still alive today, you find the exact same fundamental basal ganglia architecture.
A lamprey is basically a swimming tube with a terrifying suction cup mouthful of teeth.
It is not a complex thinker, no.
It doesn't have a neocortex like we do. It doesn't write poetry or ponder its existence.
No, but it has to decide whether to swim left, swim right, attach to a host, or detach. It faces the exact same biological bottleneck.
And inside its brain.
Inside its brain, we find a striatum populated by GABAergic medium spiny neurons. We find D1 and D2 dopamine receptors. We find a direct pathway and an indirect pathway. We find a globus pallidus.
That is just wild.
The complexity of the cortical inputs has expanded enormously over 500 million years, but the core biological engine required to resolve competition and selective behavior remains largely unaltered.
Evolution found a mechanism that works flawlessly, and it never let it go.
Exactly. If it ain't broke, don't fix it.
So how does this ancient, flawless engine actually enforce its choice? You mentioned earlier that the direct pathway is the go signal, but the way it creates that go signal requires us to invert how we normally think about brain activity.
It's totally backwards to our intuition.
Right, because we usually think of the brain like a keyboard. You press a key, an electrical signal goes down a wire, and a letter appears on the screen. It's an excitatory model.
But the basal ganglia do not work like a keyboard.
How do they work?
They work through a mechanism of double negatives. The key to understanding this is the resting state of those efferent nuclei, the GPI and the SNR.
The exits.
Right. Under normal resting conditions, when you're just sitting still doing nothing, the neurons in these output structures are highly active. They exhibit what we call spontaneous, tonic firing.
Tonic firing.
Yeah, they are continuously generating action potentials at a remarkably high rate, typically between 40 to 80 hertz.
Wait, they're firing 80 times a second, constantly, just while I'm sitting here.
Continuously. And crucially, these are GABAergic neurons. Their output is inhibitory.
So 80 times a second, they are shouting, stop, 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 to their downstream targets in the thalamus and the brainstem motor centers.
Yes, they impose a rigid, continuous, unrelenting inhibitory clamp on the entire motor and cognitive system.
I think we need a powerful analogy here because it's so counterintuitive.
Yeah.
Think about driving a car.
Okay.
Intuitively, we think that to make the car move forward, you press the gas pedal, you excite the engine. But the basal ganglia operate on a different physical model.
They do.
Imagine you are in a massive, powerful car. The engine is running high and the transmission is already in drive. But you have an enormous, heavy parking brake pulled all the way up.
I like this.
The car is vibrating, straining against the restraint, but it is clamped down. The cerebral cortex is the engine, revving with potential plans and desires. But the basal ganglia's tonic firing is the parking brake, holding everything completely still.
That is the exact physiological reality. The brake is always on.
So if the parking brake is always on, how do you move? You don't necessarily press the gas harder.
No, you selectively release the brake. This is the definition of disinhibition.
Walk us through the exact sequence of a disinhibition event. Let's say my cortex decides I want to reach for a glass of water.
Okay, the cortex sends that specific request down to the striatum, activating the D1 medium spiny neurons of the direct pathway.
The go pathway.
Right. Those D1 neurons fire. Their axons travel down to the GPI. Because the D1 neurons are GABAergic, they release a burst of inhibition onto a very specific, topographically mapped subset of neurons within the GPI.
Which subset?
The exact neurons that are holding the brake on the reach for water motor program.
So the D1 neurons inhibit the inhibitors.
Exactly. It's a double negative. The D1 input transiently silences those specific GPI neurons. Their 80 Hz tonic firing suddenly drops to zero.
The heavy parking brake on the thalamus is momentarily lifted.
That is disinhibition. The thalamus, freed from the clamp, is now able to excite the motor cortex and drive the actual reaching behavior.
Wow. So I release the brake, but only the specific brake holding back my right arm. The brake holding back my left leg is still clamped down.
And it gets even more robust than that. While the direct pathway is releasing the brake on the selected action, the indirect pathway is simultaneously engaging.
Okay.
The cortical request also excites the D2 neurons. The signal travels through the GPA, frees them the subthalamic nucleus, and the STN sends a massive excitatory burst of glutamate down onto the rest of the GPI.
The unselected parts of the GPI.
Yes. The STN causes the GPI neurons surrounding our target to fire even faster than their 80 Hz baseline.
So it clamps the parking brake down infinitely harder on all the competing actions.
Exactly. We call this a center surround organization. You precisely disinhibit the center, which is the chosen action, and you hyperinhibit the surround, which are the competing actions.
The basal ganglia function by maintaining all potential options under rigid inhibitory control and selectively, momentarily removing that inhibition only for the single option that wins the competition.
That is the core mechanism.
But how does the striatum declare a winner? We said earlier that the striatum is receiving emails from every department.
Right.
It's evaluating the urge to scratch my nose against the urge to keep paying attention to this complex explanation. Those are completely different types of desires. How does a biological circuit compare apples and oranges?
It's a phenomenal computational problem. To evaluate heterogeneous options, the basal ganglia require a common currency.
The common currency.
Yeah.
Yeah.
You cannot compare the value of an apple and an orange unless you assign them both a numerical value in a shared currency, like dollars. In the nervous system, that common currency is biological salience.
Salience, or relevance.
Exactly.
But how is salience physically represented in a neuron?
It is represented by the intensity, the synchrony, and the dopaminergic context of the cortical input.
Okay, break that down.
The cortex tags different signals with varying levels of intensity based on sensory context, emotional state, which comes from the amygdala, and internal homeostatic drives.
Like hunger or thirst.
Right. The complex microcircuitry of the striatum, the lateral connections between the medium spiny neurons, the modulatory interneurons, is computationally designed to evaluate these relative intensities.
So it's basically doing math.
It is. The option that arrives with the strongest, most highly synchronized input, bathed in the most favorable dopaminergic tone. The option carrying the highest biological value in that exact millisecond is the one that overcomes the local inhibitory networks, activates the direct pathway, and wins the disinhibition sweepstakes.
That makes perfect sense for a snapshot in time. But biological life is dynamic.
Always changing?
The environment changes. What is highly salient and rewarding today might be deadly tomorrow. We are not born knowing that typing on a keyboard is a salient action.
No, we have to learn it.
Right. If the basal ganglia select the best action, how do they learn what the best action is over a lifetime of experience?
Well, we have mapped the engine and we have seen how the brakes work. Now we need to look at the mechanic that tunes the engine. We need to shift from action selection to reinforcement learning.
And this brings us to a massive paradigm shift in how we view dopamine.
It really is.
Earlier, we talked about dopamine as a volume transmitter, a sprinkler system setting the mood of the striatum. But it has a secondary, far more dynamic role as a teaching signal.
To understand this, we have to look back at the behavioral psychology of the early 20th century. Edward Thorndike, a foundational psychologist, proposed the law of effect in 1911.
The law of effect.
Yeah. He famously put cats in puzzle boxes and observed how they learned to escape. The law essentially states that behavioral responses that are followed by a satisfying outcome, a reward, are more likely to be repeated in the future.
Makes sense.
And conversely, actions followed by a dissatisfying outcome are less likely to be repeated.
It's the absolute bedrock of animal training, and honestly, it explains a vast amount of human behavior. You do something, you get a cookie, you do it again.
Right. But for decades, it was just a behavioral observation. We didn't know the actual neural hardware that enforced the law of effect.
So when did we figure it out?
The breakthrough came in the late 1980s and 90s, largely through the pioneering work of neurophysiologist Wolfram Schultz.
What did you do?
He was recording directly from the dopaminergic neurons in the midbrain, the VTA and SNC of awake, behaving monkeys while they were performing learning tasks for juice rewards.
Okay, monkeys and juice.
What he discovered revolutionized our understanding of the brain. He found that dopamine neurons do not simply fire when you get a reward.
They don't.
No. They encode a mathematical variable that computational neuroscientists called a reward prediction error.
This is one of the most elegant concepts in neuroscience. Break down exactly what a reward prediction error is.
It's the mathematical difference between what the brain expects to happen and what actually happens. Let's walk through Schultz's classic experiment.
Okay.
You have a monkey sitting in a chair. Suddenly, completely out of the blue, the experimenter delivers a drop of sweet juice to the monkey's mouth. This is an unexpected primary biological reward.
So what happens in the brain?
In that exact moment, the dopamine neurons in the midbrain fire a massive synchronized burst of action potentials, a phasic spike. This spike releases a flood of dopamine into the striatum.
So an unexpected reward equals a massive dopamine spike. The brain is basically saying, whoa, that was way better than I anticipated.
Precisely. That is a positive prediction error. Now the learning phase begins. The experimenter introduces a predictive cue. They play a loud tone, and exactly 1 second later, they deliver the juice.
So tone, then juice.
They repeat this dozens of times. The monkey's brain learns the temporal association. The tone perfectly predicts the reward.
Right.
Now, what happens to the dopamine neurons when the juice is delivered?
Well, you would think they still fire because the monkey still likes the juice.
But they don't. When the fully predicted juice arrives, the dopamine neurons maintain their normal low-level baseline firing. There is no spike.
Wait, really? No spike at all for the juice?
No spike, because the juice is exactly what was expected. There is no error in the prediction. The reality perfectly matched the expectation.
But the spike didn't just disappear, did it? It moved.
Yes. The dopamine burst shifts backward in time. The dopamine neurons now fire their massive phasic spike at the exact moment the tone is played.
Wow.
The predictive cue has taken on the rewarding value. The brain is recognizing the earliest reliable predictor of success.
That is incredible. But here is the mechanism that proves it's calculating an error. What happens if you trick the system? You play the tone, the monkey expects the juice, and you give them nothing.
This is the negative prediction error.
Yeah.
And it is physiologically profound. You play the tone and you get the dopamine spike because the tone predicts reward. 1 second passes.
And no juice.
At the exact millisecond, the juice was supposed to hit the tongue, but the dopamine neurons don't just stay at baseline. Their firing rate dramatically drops below baseline. They enter a pause. They are completely silent.
A physical manifestation of disappointment.
Exactly. The brain is calculating, things are significantly worse than I expected.
Yeah.
So we have perfectly tuned teaching signal, a spike for better than expected, baseline for exactly as expected, and a dip for worse than expected.
But the critical question is, how does the signal actually change the wiring diagram we discussed earlier? How does a dopamine spike physically rewire the striatum to make me more likely to repeat an action?
The dopamine signal washes over the corticostriatal synapses, the exact microscopic points where the cortical emails hit the dendritic spines of the medium spiny neurons.
Okay.
And it dictates synaptic neuroplasticity, specifically long-term potentiation, or LTP, and long-term depression, or LTD.
Okay, let's say you take a novel action and it results in an unexpected reward. The dopamine spike hits the striatum.
Right. The D1 receptors on the direct pathway neurons are designed to respond to this high concentration of dopamine by initiating a molecular cascade inside the cell.
The D1 receptors are coupled to G's proteins, right? They activate adenyl cyclase, increase cyclic AMP, and fundamentally change the electrical excitability of the neuron.
Exactly. This cascade physically strengthens the specific glutamatergic synapses on the D1 neurons that were just active. It induces LTP.
So the brain is literally saying, whatever specific motor program you just executed to get this juice, physically thicken the wire so you are more likely to select it again.
Yes. But it's a dual action system.
Yes.
Simultaneously, that same high level of dopamine acts on the D2 receptors of the indirect pathway.
But D2 receptors are different.
Right. They are inhibitory at the molecular level. They couple to G proteins, decreasing cyclic AMP, so the dopamine spike actually induces LTD long-term depression, weakening the synapses on the indirect pathway.
It's brilliantly efficient. A positive reward prediction error simultaneously strengthens the go pathway and weakens the no-go pathway for that specific action.
In a negative prediction error, the pause in dopamine when a reward is withheld does the exact opposite.
It weakens the direct and strengthens the indirect.
Exactly. The sudden lack of dopamine tone firmly clamps the brakes on the unrewarded behavior. ensuring you are less likely to try it again.
Over thousands of iterations, this continuous microscopic sculpting by dopamine optimizes the action selection process.
The basal ganglia become an exquisitely tuned engine, preferentially selecting actions that maximize biological reward and suppressing those that don't.
Okay, I follow the logic perfectly for learning a new skill, but I have a practical question.
Shoot.
Does this mean every single time I tie my shoes or type my password or drive my normal route to work, my brain is actively calculating reward prediction errors? Because I certainly don't feel a rush of dopamine when I successfully tie a knot.
That is a very perceptive observation, and it highlights the functional transition from goal-directed behavior to habitual behavior.
So there's a shift.
Yes. When you are first learning a skill, think of a child learning to tie their shoes. It is highly goal-directed. It requires intense cortical focus. The child is paying attention to every movement, correcting errors, and the dopaminergic reward system is highly engaged when they finally get the knot right.
Right.
Anatomically, this early goal-directed learning relies heavily on the associative loops we mentioned earlier, specifically involving the dorsomedial striatum and the prefrontal cortex.
It takes immense cognitive effort and working memory. It is cognitively expensive, and it is highly sensitive to the value of the outcome.
Meaning, if the goal loses its value, say, you switch to Velcro shoes, you stop performing the shoe tying action immediately.
Exactly. However, with massive repetition owing time, the neural control of that specific sequence of movements undergoes an anatomical shift. It physically migrates within the basal ganglia.
It physically moves.
Functionally, yes. Control shifts away from the cognitive dorsomedial striatum and transfers over to the dorsolateral striatum, which is the core of the sensoromotor loop.
So it moves from the cognitive planning center directly into the pure motor execution center.
Yes. And once a behavior is fully entrenched in the sensoromotor loop, it has become a habit.
A habit.
A true habit is no longer driven by the expectation of an immediate reward. It is driven almost entirely by the antecedent context, the sensory trigger, like feeling the shoelaces.
You feel the shoelaces in your hands, and the motor sequence simply executes automatically, incredibly fast, and with almost zero cognitive oversight.
The basal ganglia have consolidated the action sequence so deeply that it runs on autopilot. The cortex doesn't even need to be heavily involved anymore.
It is incredibly computationally efficient.
Yeah.
It frees up the prefrontal cortex to think about other things while you tie your shoes.
But there's a catch, isn't there?
Always. This efficiency comes at a cost. Habits are remarkably rigid. Because they are somewhat divorced from the immediate value of the outcome, they are hard to break.
Right.
Understanding this rigidity and the delicate chemical balance of dopamine and disinhibition is absolutely essential for understanding our final and perhaps most profound topic.
What happens when this intricate system physically breaks down?
Exactly.
Which brings us to the clinical realities. We have spent this entire deep dive building this complex engine from the ground up. We've mapped the brakes, the parallel circuits, the dopamine teaching signals.
Along the machinery.
The ultimate proof of how a biological system works is observing what happens when you remove or damage a specific part of it.
Always.
The clinical manifestations of basal ganglia dysfunction are devastating, but they map perfectly, almost mathematically, onto the mechanical failures of the braking system we've described.
Let's start with the most widely recognized disorder of the basal ganglia, Parkinson's disease. Based on everything we've discussed about disinhibition and the dopamine volume transmission, we can now understand Parkinson's fundamentally as a failure of selection.
A failure to release the parking brake.
Yes. Break down the mechanics of Parkinson's for us. What is physically happening at the cellular level?
So the core pathology of Parkinson's disease is the progressive, selective degeneration and death of the dopaminergic neurons in the substantia nigra pars compacta.
The sprinkler system breaks down.
Exactly. The striatum is no longer bathed in that vital modulatory dopamine tone.
And without dopamine, the delicate balance between the direct and indirect pathways collapses.
Because of the receptor logic, right? Dopamine normally excites the D1 receptors, the go pathway, and inhibits the D2 receptors, the no-go pathway.
Right. As dopamine levels plummet, the direct pathway becomes sluggish, underactive, and unable to mount a strong inhibitory signal to the GPI.
So it can't release the brake.
Right. And simultaneously, freed from the suppressive effect of dopamine, the D2 neurons of the indirect pathway become wildly overactive.
So the D2 neurons inhibit the GPE, which frees up the STN, and the STN violently excites the GPI. Both pathways are now conspiring to make the efferent nuclei fire as fast as physically possible.
The tonic inhibition becomes excessive and pathological. The 80 Hz resting rate of the GPI might shoot up even higher. The parking brake on a thalamus is terminally, relentlessly pulled up.
So the cortex can send down as many glutamatergic requests to move as it wants, but the striatum just cannot overcome the threshold to disinhibit the output.
This perfectly explains the cardinal motor symptoms of a Parkinson's patient.
It explains them flawlessly. It explains akinesia, which is the profound inability to initiate a voluntary movement.
Right.
The patient wants to walk forward, the motor cortex is generating the plan, but the basal ganglia refuse to release the brake.
It's locked.
And it explains bradykinesia, the extreme slowness of movement once it finally starts, because the direct pathway is struggling to maintain the disinhibition against the overwhelming surround inhibition.
Exactly. And it explains the pervasive rigidity, the stiffness in the limbs, because the basal ganglia are constantly clamping down on the brainstem postural centers.
It is a tragic condition. Functionally, the patient's higher cognitive intent is trapped behind a wall of excessive, unbreakable neural inhibition. The brakes are stuck on.
Now, the source text contrasts this by looking at Huntington's disease, which presents almost as the exact opposite mechanical failure. It's a mirror image of dysfunction.
How does Huntington's work?
Huntington's disease is a devastating autosomal dominant genetic disorder. Unlike Parkinson's, which kills the dopamine neurons, Huntington's causes a profound progressive degeneration of the medium spiny neurons within the striatum itself.
The neurons themselves die.
Yes, but the crucial detail is the timeline. In the early to middle stages of the disease, the genetic mutation preferentially destroys the D2 medium spiny neurons.
The D2 neurons, the origin of the indirect pathway.
Yes, so the direct pathway, the go signal, is left relatively intact, but the indirect pathway, the no-go surround inhibition, is physically destroyed.
The brake lines have been cut.
As a result of losing that pathway, the efferent nuclei, the GPI, and SNR do not receive enough excitatory drive from the STN. Their tonic firing rate drops significantly below baseline. They fail to adequately inhibit the thalamus.
If the GPI isn't firing, the thalamus is constantly excited. The basal ganglia are just releasing the brake on everything, all the time, even for movements that haven't been intentionally selected by the cortex.
It is a state of excessive, aberrant disinhibition. The defining clinical result is chorea, derived from the Greek word for dance.
Chorea.
These are sudden, involuntary, chaotic, highly complex movements that randomly interrupt normal behavior. A patient might suddenly flail an arm, twist their torso, or grimace.
Because the basal ganglia are failing in their primary evolutionary duty to suppress competing motor programs.
They are allowing random, low-level cortical noise to slip through the funnel and manifest as actual physical movement.
It is a complete failure of suppression. So Parkinson's and Huntington's represent the classic motor extremes, too much brake versus no brake.
Right.
But earlier, we emphasized heavily that the basal ganglia process cognitive and affective information through completely parallel loops. The exact same D1 and D2 machinery is operating on thoughts and emotions.
It is.
So what happens when this braking system fails in the cognitive domains? This is where the source material makes a connection that absolutely fascinated me. It connects the basal ganglia to schizophrenia.
This is where the parallel loop model expands our understanding of psychiatry beautifully. Schizophrenia is traditionally conceptualized as a disorder of thought, characterized heavily by positive symptoms like hallucinations, hearing voices, and delusions, fixed false beliefs.
Okay.
For decades, psychiatry has known there is a massive dopaminergic component to the disease, primarily because the most universally effective antipsychotic medications are, at their core, D2 dopamine receptor antagonists.
They block the D2 receptors, the very receptors that initiate the indirect no-go pathway.
Exactly. But the conceptual leap is hard. How does blocking a dopamine receptor deep in the basal ganglia stop a patient from hearing a voice that isn't there?
Yeah, how does that work?
We can bridge that gap through the lens of action selection applied to the cognitive and associative territories of the basal ganglia.
Okay.
In a healthy brain, the associative loop running from the prefrontal cortex through the caudate is constantly selecting the most appropriate thoughts to hold in working memory, while actively suppressing irrelevant, intrusive, or bizarre thoughts. It filters the cognitive noise.
Just like the motor loop selects a purposeful reach and suppresses a random twitch.
Precisely. The prevailing hypothesis is that in schizophrenia, there is a profound dopaminergic dysregulation, often characterized as hyperdopaminergia in the striatum.
Too much dopamine.
Right. This excess dopamine overactivates the direct pathway and overly suppresses the indirect pathway within the associative and limbic loops.
So the cognitive brakes fail.
The cognitive basal ganglia fail to adequately clamp down on non-selected competing cognitive options or internal sensory representations.
Wait, so a hallucination is essentially a cognitive twitch.
That is a brilliant way to phrase it. A hallucination, in this framework, is an internally generated sensory memory, or a fragment of an internal monologue that should have been suppressed by the indirect pathway.
But because the dopamine system is dysregulated, the basal ganglia erroneously flag that internal noise as highly salient.
They release the cognitive brake, the unsuppressed thought escapes inhibition, loops back up to the cortex via the thalamus, and intrudes into conscious awareness with the full force of a real sensory experience.
That is staggering. It is an unsuppressed cognitive intrusion, perfectly analogous to a choreic movement escaping into the motor system,
Exactly.
unifying a physical motor tremor and a complex auditory hallucination under the exact same fundamental circuit logic. The brain is failing to apply the parking brake. It's just failing in different parallel loops.
It's all the same hardware.
What about other conditions defined by behavioral intrusions, Tourette's syndrome and ADHD? They must fit this model too.
They map onto the circuitry exceptionally well. Tourette's syndrome is characterized by tics, involuntary, repetitive, stereotyped movements or vocalizations. Rather than the global breakdown seen in Huntington's, Tourette's can be viewed as highly localized, aberrant disinhibitions within specific focal channels of the sensoromotor or associative loops.
The basal ganglia momentarily release the brake on a highly specific, complex motor program, like a throat clear or a facial grimace, when it absolutely shouldn't.
Yes. Similarly, the hallmark hyperactivity and profound distractibility seen in attention deficit hyperactivity disorder can be conceptualized as a failure to maintain surround inhibition.
So the cognitive loops in ADHD are unable to maintain rigid inhibitory control over irrelevant external distractions or internal impulses. The engine cannot hold its focus on just one salient target. It keeps releasing the brake for every shiny object that crosses the visual field.
It is a deficit in the continuous suppression of competing stimuli. Now let's look at the absolute flip side of that coin. Now, we've talked about what happens when the surround inhibition is too weak and everything intrudes.
Okay, what's the flip side?
What happens when a single behavioral option becomes pathologically dominant? When the engine's selection mechanism gets stuck in a feedback loop and it can only focus on one thing. This brings us to obsessive-compulsive disorder, or OCD.
OCD is characterized by obsessions, which are deeply intrusive, distress-inducing thoughts, and compulsions, which are repetitive physical or mental behaviors the patient feels driven to perform to alleviate the distress of the obsession. How does the basal ganglia get trapped in that cycle?
In the basal ganglia framework, OCD is viewed as a disorder where the core selection mechanism is fundamentally hijacked by an overly salient input. The direct pathway for a specific anxiety-provoking thought or a specific compulsive behavior becomes hypertrophied and pathologically dominant.
The signal coming into the striatum is just too strong, and the basal ganglia can't filter it out.
Exactly. For complex reasons involving hyperactive cortical input from the orbitofrontal cortex and aberrant local plasticity within the striatum, a specific channel in the associative or limbic loop carries a massively disproportionate amount of salience.
Ah, salience again.
Because the basal ganglia are designed to always select the most salient option, they preferentially and repeatedly select this pathological option. They disinhibit the obsessive thought over and over again while simultaneously using the indirect pathway to suppress all other viable, healthy behavioral alternatives.
The patient is functionally trapped in a rigid, repeating, high-stress cognitive and behavioral loop simply because their internal selection engine is flawlessly executing its algorithm on distorted, hypersalient data.
It's a terrifying thought. The selection mechanism is working perfectly, but it's working against the patient.
And finally, we have to talk about addiction. This ties directly back to our deep dive into dopamine, the reward prediction error, and the transition from goals to habits. How do external chemicals, drugs of abuse, so completely hijack this elegant survival system?
Addiction is perhaps the most direct molecular hijacking of the basal ganglia's core machinery that exists. Think back to the reward prediction error.
Right, the monkeys and the juice.
The dopamine system evolved over hundreds of millions of years to evaluate natural biological rewards, finding a high-calorie food source, securing fresh water, successful social interaction. When we achieve those things unexpectedly, we get a dopamine spike which strengthens a direct pathway, eventually turning those survival behaviors into robust, unconscious habits.
It's how evolution ensures we keep doing the things that keep us alive.
Drugs of abuse, whether we are talking about cocaine, methamphetamine, opioids, or alcohol, short-circuit that entire evolutionary process. They bypass the normal sensory pathways and directly artificially stimulate the dopaminergic neurons or manipulate the reuptake of dopamine in the striatum.
They don't just trigger the teaching signal, they flood the engine with it.
They provide massive, unearned, purely pharmacological dopamine spikes in the ventral striatum. And these spikes are orders of magnitude larger and last significantly longer than any dopamine release produced by a natural reward.
But here is the most devastating part regarding the prediction error algorithm. With a natural reward, once you learn the cue, the dopamine spike goes away. The prediction error drops to 0.
Because the juice is expected.
Right. But drugs are chemicals. They force the release of dopamine regardless of expectation.
That's the trap.
Yeah.
Even after the addict fully expects the drug, the pharmacological nature of the substance still forces a massive dopamine spike every single time it is consumed.
So the prediction error never drops to 0. The brain is constantly, perpetually calculating. This is the most incredible, biologically vital thing I've ever encountered. Keep learning this, keep strengthening this connection.
The basal ganglia are literally tricked at the molecular level into believing that acquiring and consuming the drug is the single ultimate evolutionary survival goal of the organism. These relentless, massive dopamine signals rapidly and violently strengthen the direct pathway for drug-seeking behaviors. The behavior shifts with terrifying speed from goal-directed taking the drug to feel a euphoric high to a deeply ingrained, rigid, unconscious habit controlled by the sensorimotor loop.
And once it's a habit in the sensory-motor loop, it's divorced from the actual outcome.
The individual continues to seek and consume the drug compulsively, driven by a rigid, artificial habit, even when the drug no longer provides any pleasure, and even when it causes profound, catastrophic negative consequences to their health, their relationships, and their life.
The very reward learning mechanism designed to keep the organism alive has been weaponized to destroy it.
It is genuinely chilling when you break it down functionally like that.
To try and summarize this immense clinical landscape we've just covered, what we see is that wildly different conditions, things that seem to have nothing in common on the surface. From the physical, frozen rigidity of Parkinson's to the chaotic physical flailing of Huntington's. From the terrifying, intrusive thoughts and voices of schizophrenia to the repetitive motor tics of Tourette's.
From the trapped, anxious loops of OCD to the absolute, compulsive, life-destroying drives of addiction.
All of these human experiences stem from the exact same underlying circuit logic. They are all, at their core, mechanical failures of the basal ganglia's braking and selection system. They are just applied to different parallel loops, motor, cognitive, limbic, and caused by different specific molecular breakdowns, loss of dopamine, loss of MSNs, hyperactive receptors. It is an incredibly beautiful unifying theory of brain function and dysfunction.
It is the power of understanding the underlying architecture. It demonstrates that the brain does not invent a brand new bespoke circuit for every different problem it faces. It iterates on a deeply conserved, fundamentally brilliant architecture. The basal ganglia are the universal deciders.
Well, let me take a breath and recap the incredible journey we have been on today. We started by mapping out the dense, intricate cellular anatomy of the basal ganglia.
Hardware.
We looked at the massive receiving inbox of the striatum, populated by those crucial D1 and D2 GBAgic medium spiny neurons. We identified the intrinsic regulatory middleman, the GPE, and the broadcasting efferent nuclei, the GPI and essinger.
We explored the complex wiring diagram, understanding how the direct pathway acts as the targeted go signal, while the indirect pathway acts as the broad no-go signal, and how the hyperdirect pathway acts as the emergency brake.
We looked at the macro architecture of the parallel, segregated loops that process motor, cognitive, and emotional data using the exact same algorithmic software. We dove deep into the evolutionary necessity of a biological brake system, tracing it all the way back to the jawless lamprey.
We learned that action selection relies not on pressing an accelerator, but on selectively disinhibiting a desired action while clamping down fiercely on all the alternatives.
We saw how dopamine acts as a profound, dynamic teaching signal, physically rewiring the corticostriatal synapses based on mathematical reward prediction errors, transforming novel goal-directed actions into rigid automatic habits.
And finally, we saw the profound, often devastating consequences when this elegant system breaks down across a wide spectrum of neurological and psychiatric disorders.
It is an immense amount of material to process in one sitting. It requires a fundamental shift in how you visualize neural activity.
But it is foundational knowledge. Understanding the basal ganglia is the absolute key to understanding how we transition from internal thought to external action.
Which leaves us with a final, slightly provocative thought for you to consider as we wrap up. We talked a lot today about unconscious habit formation, the common currency of salience, and the constant violent suppression of competing impulses.
I know where you're going with this.
I want you to think about your own behavior right now, in this exact moment. The very fact that you are sitting still, that you have continued to listen to this dense, highly complex neuroscience lecture for the past hour, means something incredible is happening inside your head.
It means your basal ganglia are actively, right in this very millisecond, firing at 80 hertz to suppress the urge to check your messages, the urge to stand up and stretch, or the urge to let your mind wander to what you're going to eat for dinner.
Exactly. Your internal dopaminergic system has calculated that the reward of understanding the brain, of grasping this complex architecture, possesses a higher biological salience than any other competing action available to you right now.
Your direct pathway has successfully disinhibited the complex cognitive act of listening and processing, and your indirect pathway is holding the line, clamping down hard on 1,000 other biological distractions.
Your basal ganglia are working perfectly.
A beautiful demonstration of the system operating flawlessly in real time.
It really is. Remember, this deep dive is just one piece of the puzzle in our ongoing exploration of the neuroscience of movement. We will absolutely be building on these concepts, the loops, and the dopamine dynamics in the chapters to come. Until next time, keep exploring the machinery of your own mind.